To import these data, you can follow the standard RT-PCR workflow in Array Studio, or you can run modules from the menu. First, open Array Studio and create a new local project. If you have access to an Array server, you could create a server project. This allows you to easily share data and views with colleagues, and the analysis steps can be performed on the server. For information on uploading data for server-based projects, see the Getting Started with Server Analysis video tutorial. For this RT-PCR experiment, we will create a simple project. Now let's add our RT-PCR data to our new project. Click Add Data, Add Omic Data, Add RT-PCR Data. We will use the Import RT-PCR Wizard. This wizard will carefully walk through each step to ensure proper importing of the RT-PCR data. The tutorial data are tall skinny, so choose this option. If your data are in a matrix with either one sample or one gene per row, you could choose a different option. Our measurements are CT values, but if your measurements are abundant, uncheck this box. Now click Import Data. Our data are in an Excel file, but it can also be a tab delimited or comma delimited file. We will choose the tab containing our data. Now click Next. Now choose the metadata and sample data columns. We could add optional metadata from this sheet, but we will just add it from annotation and design tables later. Now preview the data. In the heat map table, look for any yellow values, which indicates missing data. We can attach additional design and annotation metadata to our sample data from separate files or from tabs of an Excel spreadsheet. In this experiment, we have two tissue types, actually two cell lines, and a treatment and control group for each tissue type. We don't have any technical replicates in this data, but if you do, you can choose to combine or remove replicates that you would like to exclude. If you have missing data, you can choose to replace those data with a very low value, such as 35, which is a very low abundance value in CT scale. The tutorial data are CT data. We want to calculate delta CT. The wizard will return log transformed relative abundance. The wizard contains several other options for different transformations. We will now normalize the data. We could normalize against one or more housekeeper genes in the data, but we will just perform a global normalization. You can inspect the normalizer values, now we can preview our data. Although the data were initially in tall skinny format, they are now in a matrix format, with each gene listed as a row and each sample listed in the columns. Your new data are omic data type. The table contains the values, and metadata are attached as annotation and design tables. You can rename the omic data objects. For example, if you need to import more RT-PCR data, you might want to rename your data to something more meaningful than RT-PCR data. In our annotation table, the column called Gene ID is actually ensemble IDs. We can change this, but not while it's set as the ID column. Let's first change this column name to Gene Symbol. Now right-click on the annotation folder and choose Set ID Column to Gene Symbol. We can now change this column title. Gene Symbol is now set as the ID column, but we can switch it back to Ensemble ID. You can switch the column and row IDs in your data to any other column in your design or annotation table as long as they are unique.